Hello. Welcome back to the Physionic podcast. My name is Nicholas Verhoeven. I am a doctoral candidate in molecular medicine, and I'm a cell biology researcher. And actually, some of the research that I do is related to the topic of today's podcast. But before I get into the podcast, if you haven't seen my brand new video that came out looking at over 100 studies on how the ketogenic diet or low carb diets in general affect our blood cholesterol levels. And I'll have one on body weight, triglycerides or blood fats, and blood sugar and insulin. Those will come out in the coming weeks, but the first one on cholesterol has released. So if you haven't checked that out and you are considering a low carb diet, then definitely jump onto that video. I think it's only 9 minutes long. It burns through over 100 studies, as well as explains the physiology, explains the results, why the way that they are the way that they are, conclusions, takeaways. It's if I may say, it took a lot of work over several months to read all those studies, the 13 studies that I individually read, and then the meta-analysis on top of that, adding several or many more studies, like 100 more studies to the mix as well. So if you're interested, please check that out. Certainly trying to promote that as much as possible just because of the sheer amount of work that I put into it. Okay, but that is not a part of this conversation. This conversation today is going to be discussing mitochondria, and not the traditional sense of what we think of when we think of mitochondria. Obviously, the most common is the zombie-like conversation starter that people like to throw in there when I say that I work at least partly on mitochondria. They'll say the powerhouse of the cell, and you've heard that a billion times, and I'm sure you'll hear that a billion more times. And that's not untrue. That is absolutely not untrue. I'm not here to debunk if mitochondria provide cellular energy. They most certainly do. However, they do a lot more than that, and that is what this podcast is about. It's going to elucidate a different activity or a different function of mitochondria, and that function is to kill your cells. As weird as that sounds, on one hand, the mitochondria giveth, and on the other hand, mitochondria taketh. Giveth energy and taketh by killing the cell. So why would mitochondria want to kill our cells? And that raises three different points that I'll be discussing. So first is, why do cells die? Why do we want cells to die? That's the second one. And then how do mitochondria actually contribute to this process? So let's go ahead and jump in. Of course, I've got visuals for anybody that wants to see visuals of what this process looks like. So we're going to get into the molecular details, and then I'm going to actually show you some real images of what's happening to the cell as the mitochondria is doing that. So should be cool from a researcher's perspective. Hopefully you enjoy it. And if you do, certainly let me know in the comments and leave a like and all that good stuff. All right, let's do it. Why do cells die? Well, they die for many reasons. One of the most obvious is that they age out. So, for example, red blood cells last about 3 to 4 months, and then they get more and more stiff. Their cell membrane gets more and more stiff, and then eventually they're supposed to be really flexible and have the ability to move through these different capillaries, which are really small tubes in our body, and sometimes they can't because they're so stiff. So they end up breaking. The cell membrane ends up breaking, and you get the release of everything that's inside the red blood cell. That leads to the cell dying, or that red blood cell can end up at the spleen and can also be destroyed in the spleen as well. So they're old age. That's a great reason why cells have to be destroyed and then recycled by the creation of new cells. Another huge one is cancer, right? If you have the uncontrolled proliferation, the uncontrolled division of cells, that is obviously a negative. So if a cell starts to become more cancerous, then the cell has its own mechanisms, as well as the body has mechanisms to make sure that that cell is ultimately destroyed before it can actually turn into a really cancerous cell. To prevent infection. I have a lot of content that's going to be coming out probably in the next 2 to 3 weeks, if I can get my video editing done. That is going to be explaining this mechanism in some great detail, some incredibly great detail. I'm really excited. It's going to be a series called The Stories of the Immune System, or something, or the Stories of Inflammation, something along those lines. And it actually describes exactly this. It describes how our immune system destroys cells or fights off infections. And one of the ways is by the destruction of cells that have been already infected with, for example, a virus. So that is definitely a huge one. Another one is to form our bodies. I'll actually discuss this a little bit more in the next question or in the next section. But really, when we're developing as a fetus, and even as babies, but certainly as a fetus, our cells are constantly killing themselves to allow us to have shape. So if we have a particular shape at, let's say, certain weeks in the pregnancy, and then that shape obviously has to change. We grow, certainly, but we also have to kill off certain cells to make sure that the shape of our body turns out the way that it does. And that is done through some of the mechanisms that I'll be describing here. Another one is, and the final one that I'll discuss just briefly here to answer the question, is to prevent autoimmune diseases, which is another one that I'm going to be creating a really, really cool video on, looking at how cells essentially create mass genocide, where they kill themselves. I mean, we're talking millions and millions of cells end up killing themselves to make sure that they don't end up attacking your own body. Now, if you're intrigued, hit the subscribe button, because that video will release in a number of weeks as well. And that's really going to describe what that exactly means. But it's essentially overall to avoid autoimmune diseases. Okay, so why do we want cells to die? Well, obviously to prevent disease, as I just mentioned, but really to get a little more specific about how our body's molded. Have you ever thought about how your fingers get created? Well, look at them, or your toes. Really, these fine details, these grooves between your fingers are generated. At one point, it was all one globular structure. And as you developed in the womb, these cells that lined your fingers ended up dying to create these actual finger-like projections. So they're obviously cell death is incredibly useful for our own development. So that's kind of the big point. But we also want this process to be controlled. We don't want this to just occur. A great example of this is with a condition known as rhabdomyolysis, which if you're really familiar with working out, or if you're familiar with David Goggins, he's someone who has suffered from rhabdomyolysis in the past. And really it's a condition where, and I actually have a detailed video and article on rhabdomyolysis. So if you're interested, check that out. Just type in Physionic rhabdomyolysis into the YouTube search, or into search in general, and you'll find it. But essentially, it's where you work out so hard and just push your body so hard within a single session, or even in repeated sessions, that your muscle cells end up dying. So the actual cells themselves will end up incurring so much damage that they can't overcome that, can't recover, and therefore they end up dying. And ultimately, there's a lot more complexity, because the real issue is what leaks out of the muscle cells to then cause further damage and could potentially be life-threatening. So in that situation, we're talking about necrosis, which is a particular type of cell death. And I'm going to show you some images now of what that looks like, just so that you can have a little bit of an idea. So these aren't muscle cells, just to be clear. Muscle cells are very linear. But here we've got I forgot exactly what kind of cell this was when I pulled it off. But this is a normal cell, so it's perfectly functional, no problems with it. And then we've got a necrotic cell here, where things are essentially starting to kind of fall apart. Everything's starting to break apart. And that may not look all that different from an apoptotic cell, which is programmed cell death. So this is a controlled process, and this is normally considered an uncontrolled process. So this actually occurs to the cell, and this tends to occur, while it can occur to the cell technically as well, this is typically designed this way. So, and this is the process that I'll be describing to you. We're going to look at the actual molecular mechanisms that cause that through mitochondria. But necrosis is just the slow, just overall, just kind of falling apart of the cell. Now, apoptosis, however, has particular molecular mechanisms that lead to what's called blebbing, where you can see these pieces of the cell are slowly breaking off and are eventually going to be consumed by the immune system, or particular cells of the immune system. But again, it's controlled. That's really the big point that I'm trying to get across here. And there are other types of cell death as well. It's not just apoptosis and necrosis, although they're the two that are probably talked about the most. There's also entosis, which is actually one that I just learned about today. I was doing a little bit of background reading for this, reading a few reviews and for this podcast. And entosis is such a wild one. I mean, it's really a wild one because what it is essentially a cell engulfing another cell, or two cells engulfing a single cell, and then that cell is stuck inside of another cell. And then the engulfing cell, ends up bombarding it with a bunch of destructive enzymes and stuff to destroy that cell. Now, however, there are cases in entosis where the engulfing cell will engulf the other cell. And this is for any immunology people. It's actually different from, through a different mechanism from phagocytosis. But let's just say you have the engulfing of another cell. That engulfed cell, so not the engulfing cell, but the engulfed cells, the one that's been taken in, can sometimes escape. I mean, how crazy is that, to have this cell that's just, it's almost like a gluttonous cell. It just comes over and it's just like, I'm going to eat you. So it just absorbs the other cell, and then the other cell, if possible, can actually escape from being consumed. Not that it's really being consumed. It's kind of a weird process, but I just thought that was super wild. And then there's also autosis, and then there's a few other mechanisms as well. But autosis, if you're familiar with autophagy obviously is something that's such a hot button topic. That's the other aspect of what I study, mitochondria, apoptosis, and autophagy. But autosis is autophagy inside the cell to such a degree that it ends up killing the cell, which can also happen. And if you're worried about your fasting, if that jumps into your mind, well, am I fasting too much and it's causing autosis? That's probably not an issue. This typically happens in pathological states. Okay, so the big point here is that you want controlled cell death. You don't want uncontrolled cell death. All right. So now let's round this into, how do mitochondria contribute to this process? There are processes, and I'm going to be focused on apoptosis, which is one of the ones I showed earlier, but there are there's a few different ways, not a whole lot, but a few different ways that can lead to apoptosis. You can have an immune cell, or any cell, but more so an immune cell, that will bind to the cell that needs to be destroyed, and it will convey a series of molecular signals to that targeted cell that will then lead that targeted cell to then destroy itself. However, there are other mechanisms through things like cell stress, which are mediated through mitochondria. So let's look at that. Okay, so here I've got a picture of a cell membrane. This is a cell membrane. It's square. Let's just ignore that, although there are certain cells that are square. We will just, I'm trying to talk about this as simplistically as I possibly can, while still offering you a decent amount of detail on the molecular mechanisms here. So we have cell stress, if that's through like oxidative stress, or you get some sort of signal, an intramolecular signal, essentially proteins and fats and things like that have a particular shape will be damaged or whatever it might be, and that ultimately leads to an increase in a family of proteins. This isn't just one protein, but have it labeled here as a family of proteins called the BH3 proteins. So cellular stress, immense cellular stress occurs. Then you have a rise in the BH3 family proteins. And then from there, those will influence or activate two particular proteins that we're most interested in called BAX and BAC. And you can think of them like troublesome brothers and sisters. So you've got BAX is the sister and BAC is the brother. And they are typically known as monomers, meaning that they are single proteins. And once activated, however, they will what's known as oligomerize, where they will join together and create a complex of proteins. So multiple of these proteins will come together and create a pore in the mitochondrion. So the mitochondrion has two membranes, kind of like the cell membrane has one membrane. The mitochondrion has two membranes, the outer membrane and the inner membrane. And this BAC-BAX complex that is activated by BH3 proteins that then oligomerize will insert themselves into the outer membrane of the mitochondrion. Now that ultimately then leads that you have an opening between what's called the cytosol, which is the inside of the cell, but not the outside of the cell, the inside of the cell, and the inside of the mitochondrion. So suddenly there's no barrier between them, and therefore the inside of the mitochondrion, especially between these two membranes, can then leak out. So that's exactly what we end up seeing. So you get the leakage of the mitochondrial proteins from inside the mitochondrion to the cytosol of the cell. Now the cell will actually detect that, and it does so through, there's a particular protein, there's a series of proteins that come out of the mitochondrion in this situation, but there's a particular protein or complex known as cytochrome C, and that will actually interact with another protein inside the cytosol known as APAF. And these two proteins, and there's a lot more complexity to this, but these two major proteins will ultimately end up generating what's known as the apoptosome. I had to say that somewhat dramatically because it looks kind of dramatic. And yes, it does look like this kind of this multi-layered structure. And the apoptosome is, if you can tell by the name, it's apopto, right, which is very similar to what we talked about earlier, apoptosis. So cell stress increases BH3, oligomerizes BAX-BAC. That creates a pore, mitochondrial proteins release, and then you get the generation of the apoptosome. Now the apoptosome then will cleave or cut what's known as a procaspase, which is a protein that has a particular function. I'm going to discuss that function in just a second. But the cleavage of the procaspase leads to the generation of the caspase, so caspase 9 in this situation. Now, procaspase is an immature, inactive, largely inactive, for this case, inactive form of a protein. And then that gets cleaved by the apoptosome to generate caspase 9, which is an active form of the caspase proteins. Now, caspase 9 has a unique function in that it also activates two more caspases. It activates caspase 3 and caspase 7. Again, two other proteins that belong in the caspase family, but are obviously labeled differently because they have slightly different functions from caspase 9. Caspase 3 and caspase 7 then are called terminal or destructive caspases, or kind of majorly destructive caspases. And what they end up doing is they end up going through the cell and just destroying everything. They just start wreaking havoc on everything. So they'll destroy hundreds of different types of proteins within the cell. Now, think about that in the context of cell death. probably not the greatest thing for the cell if you have these two proteins that have now been activated and they go around just destroying all kinds of different cellular proteins that are necessary for the cell to function. It's kind of like going around in your house and just dousing it all in gasoline and then caspase 7 coming around and just throwing a match on it and everything catching on fire. Probably not the greatest thing for your house. So it's sort of similar in that way, except I guess technically caspase 3 and caspase 7 are going around with hatchets and axes and chainsaws and just destroying everything in your house. So that ultimately leads to such traumatic damage that the cell ends up dying. Now, what does that look like? Now, I showed you a 2D or sort of 2D representation here, but let me show you a 3D, which I think is really cool. This is a 3D representation of what's going on. So a normal cell on the left side here, and then you have the apoptosis blebbing that's occurring. So you can see that it looks really effed up. And that's because these caspases are inside this cell and are just destroying everything. And then you've got a bunch of other cellular mechanisms that are essentially succumbing to apoptosis. And that ultimately leads to the generation of this dying cell. And it doesn't last that much longer after that point. Okay, so there you have it. The conclusions, takeaways for anybody that ended up skipping to this section, which it would be a shame because you just missed all the images and some pretty cool stuff, if I may say so myself. Okay, so in conclusion, essentially mitochondria can contribute, are used for programmed cell death to fight cancer, fight infections, to form parts of your body. Remember when I was talking about the fingers, among other reasons, to ensure a healthy overall body by the sacrifice of some cells. So some cells have to be sacrificed for the overall you to exist and flourish in a healthy manner. Okay, folks, that's what I've got for you. Hopefully you found this fascinating. And if you'd like more of these kind of molecular dives on how the body functions, how the cells function, the cell biology, the molecular biology, then certainly let me know. And with that, thanks for stopping by as always. And I always appreciate you. And I'll catch you in the next one. Bye.